Everyone, my name is Ellen Koch, and I'm a PhD candidate at Lynn Raymond's lab at UBC. And today I'll tell you about my project that I've done as part of my Canadian Open Neuroscience Platform scholarship. I was a recipient of the 2020 CONP Scholar Award, and my co-advisor for this project was Eric Ittrey at Carnegie Mellon University in Pittsburgh. So my project centers around Huntington's disease, which is a neurodegenerative disease that leads to movement, cognitive, and psychiatric disturbances. And the main site of neurodegeneration in Huntington's disease is the dorsal striatum. I'm using a technique called fiber photometry to measure activity of the dorsal striatum in mouse models of Huntington's disease. So I'm using fiber photometry to measure activity of the Gcamp calcium sensor while mice perform behavioral tasks, such as the accelerating rotor rod and the open field task shown here. And the mouse model that I'm using is called the YAC128 mouse model. So with fiber photometry, we can measure activity of fluorescent sensors during behavior. And in this case, this mouse is performing the rotor rod motor learning task, and we're measuring Gcamp activity in the dorsal striatum at the same time shown here. Here's an example of the activity before, during, and after an accelerating rotor rod trial. And you can see how the activity has an initial increase and that the activity looks different while the mouse is doing the trial compared to before and after. I also have an example here using the GluSniffer glutamate sensor as a way to measure glutamate release during this task in another mouse. So these are some examples of the kind of data that you can collect using fiber photometry. Another analysis that I'm doing is using DeepLabCut, which is a machine learning tracking software to track the position of the mouse's paws during the rotor rod task. And this is the way that I can look more closely at this behavior. Here's two examples, the top being a wild type mouse, and the bottom is a Huntington's disease YAC128 mouse. And on the right side, I'm showing a graph of in red and blue of the right and left paws over time. So you can see that over time, their steps are getting larger and larger, and they're actually slipping more where they almost fall off the rotor rod. And in the case of the YAC128 example, you can see that they're slipping much more often than the wild type, and they're just overall less coordinated compared to wild type. So using DeepLabCut, followed by my own analysis in scripts that I've written in Matlab, I can extract many different measures, including looking at the paw position, foot slips over time, height, variance in height, and I can look at these kinds of measures over a single trial shown here, or over days of training. And using this methodology, I found some very interesting differences between the genotypes where the Huntington's mice have many more foot slips compared to wild type. And by the way, these are early stage YAC128 Huntington's mice who actually are performing well on the rotor rod when you just look at latency to fall. So even though they seem like they're performing just as well as wild type, when we look closer at their behavior using this analysis, we can see that they're impaired when we look at their paw kinematics. I also found some differences looking at the paw height at 6 to 7 months of age, an older age group in these mice, as well as a deficit in the standard deviation of the paw height. And I've also looked at the open field data using DeepLabCut and Matlab, as well as using a machine learning software developed by Eric Ittrey's group called VSide. And using these techniques, I can look at normal sort of standard analyses, such as total distance traveled and center time. As well, using VSide, I can look at specific behaviors that the mice are engaging in. And I found, using this analysis, some interesting differences in the types of behaviors that the wild type versus the Huntington's mice engage in the open field. Lastly, for the Gcamp data itself, I've analyzed this data in various ways. I can look at the average delta F over F and Z score values using my script. I can calculate the peak frequency, amplitude, and width of all the individual calcium events. And a new analysis that I've started to do that I'm very excited about is actually aligning the data to specific behaviors. So down here, I'm showing an example of the Gcamp activity aligned and averaged over all of the foot flips that this mouse has on the rotarod. And you can see this large increase in activity following the foot flip, or the average foot flips in this mouse. And I'm also going to be uploading data from 2 color photometry experiments as well. So all of this data is already or will in the future be shared on these online platforms shown here. So raw and partially analyzed data will be available on the Open Science Framework. And also I'm planning to also put this on the CONP portal too. GitHub is where you can find my analysis scripts as well as sample data. And the Dataverse project is where published data will be uploaded as well. And I just wanted to include a few links here. So I have scripts and sample data available online for rotarod behavior analysis, open field analysis, and photometry analysis. And there'll be more available soon. And this is on the UBC Dynamic Brain Circuits GitHub. So with that, I would just like to thank everybody on this slide and many more that I probably have not included here, as well as the funding sources, especially the CONP, for making this work possible. Thank you.